My name is Christoph Alexiou. I'm a physician and nanomedicine researcher in Erlangen, and I'm very happy to announce the first talk from Dr. Camille Peitsch concerning world's smallest glycolipid-based nanodrug against neurodegenerative diseases, a journey to clinics and beyond. So Dr. Peitsch, the floor is yours.
Good morning, everyone. Thank you very much for this nice introduction. I'm Mr. Alexiou. Yes, today I will tell you about Talineuren. I've presented Talineuren, I think, like 2 years ago during the last CLINAM summit. That was also virtual. And today I would like to tell you a bit more about the results and the upcoming projects for Talineuren, and start with Talineuren as such. Then the relevance for Parkinson's disease. I'm really happy also to show you the new data on amyotrophic lateral sclerosis that we could gather with the product. And small overview of the patient access pathway that we are pursuing, and a conclusion at the end. So as a refresher for the one who already saw the presentation 2 years ago, Talineuren is a liposomal GM1 ganglioside solution. It contains 180 milligram of GM1 ganglioside. It's a white translucent product, as you can see on the picture on your left. It's stable for over 9 months. It's a sterile concentrated solution for IV or oral application. And the nice thing about it, it's exclusively made of body-owned molecules, which we have made a lot of biodistribution assays and different assessments in mice and rat models. So one was a quantitative analysis of the GM1 levels in the brain in nanogram per ml of total brain. We have compared Talineuren per os versus IV compared to the free micellar GM1 IV. And in that context, we could see that with the Talineuren IV, we can double the amount of GM1 found in the brain as such, as a whole total. But it was not significantly really an upgrade when it was administered per os. However, on brain slides, the label Talineuren could be really nicely recognized by the labeling of the fluorescent red dye DIL, which is typically used on brain slides, which here you can see the dopaminergic, there was a colocalization analysis of the dopaminergic neurons through the tyrosine hydroxylases. And that leads me to the relevance for Parkinson's. So one of the last big clinical trial that was conducted with free micellar GM1 was concluded in 2013 up to 2015 by Jay Schneider at the Thomas Jefferson University in the US. It was a randomized controlled delayed start trial where patients got a daily dose of 100 milligram, 200 milligram, twice a day, 100 milligram injected subcutaneous. As you can see on the graph, it was a successful study. It lasted 2 years of treatment, and then there were follow-ups up to 4 years. And you can clearly see on the blue line, you have like the treatment we're starting with a GM1, the free micellar GM1. And you have the delayed start that was started a few months. The treatment was started with the GM1. And then at the end of the treatment, you could also see that beside to the placebo, you can have an effect of the free micellar GM1 compared to the reference group on the motor score of the UPTP, of the universal scale UPDRS. So, you know, Lidin et al. Have made quite some studies over the past 20 years and even more on GM1 gangliosides, and especially also in correlation with the deficits of GM1. And yes, in Parkinson, we mainly talk about alpha-synuclein aggregates. We talk about reduction of dopamine levels and dopaminergic neurons. But one thing that is striking and has been proven in animal models, but also in the analysis of postmortem analysis in humans and also made by biopsies in other studies is that you have a deficiency of GM1 gangliosides, not only in the central nervous system, but also systemic in other tissues. And maybe another aspect is that the free GM1 ganglioside is already in use for Parkinson's disease in China and Brazil. So our first clinical trial is a safety evaluation with the liposomal GM1. So now we are talking about talineuren, and this trial is ongoing. We started in December 2021 with a 14 weeks dose escalation with a total number of patients of 3. And currently we are reaching the end of the dose escalation where 2 patients already got the 720 milligram dose and 1 patient just got the 660 milligram dose actually yesterday. We have then as a follow-up a dose consolidation with over 8 weeks for 9 patients where the maximum suitable dose will be discussed in the coming weeks. And there will be in the end a follow-up of about 4 weeks for foreseen end of the full clinical trial by the end of August 2022. The primary outcome for safety evaluation are the occurrence of the adverse events as well as the severe adverse events. Several routine labs have been also put in place for security and safety reasons, as for example the anti-GM1 antibodies that could be a risk for that therapy. The results of this are not yet available, as well as the apolipoprotein B, especially due to the amount of lipids that are administered. The secondary outcomes are mainly efficacy-related endpoints, amongst other the MDS UPDRS scores. And the recruitment went very well, so we are already booked for the whole clinical trial. This one, it is taking place in Bern in Switzerland. And so far, so as of today, we have a very nice tolerability and no severe adverse events were reported until now. And I think a very nice sign of this very well ongoing clinical trial is that a patient requested to actually pursue the therapy. And we made this request by Swissmedic, and this one was already approved. So patients can pursue the therapy after the end of the clinical trial. So after like August is coming soon, so we are already saying, okay, we need to start the efficacy study, the phase 2. How do we want to do this? So we will do a double-blind randomized placebo-controlled efficacy study as an add-on therapy to stable patients with L-DOPA. The study powered will be actually based on the NEON trial outcome. So this is not yet available as an information, and the dose as well. We will decide this upon the dose consolidation of the NEON trial. We expect to have a start in Q4 2022, and our lead principal investigator and coordinating will be Michael Schipper, that you can see on your right. He also has been now being the principal investigator of the NEON trial. So this LEAP study will be a multi-center study, and we really look also forward to see more of the efficacy of talineuren. For amyotrophic lateral sclerosis, we have been now working very intensively for reaching a whole mode of action and try to understand talineuren in this specific disease, because it's another disease even. And interestingly, I would like to show you a few results. So it's a rare disease. It's a very tragic life expectancy of about 3 to 5 years. And the current therapies that are available are edaravone and riluzole, which actually extend the life expectancy of about 2 to 6 months, maybe a bit more, but it's are also bound with very bad side effects. The ILS is actually also a sporadic disease to 90%, and the 10% of genetics. We have shown that we can also, by the biodistribution, find talineuren in the ventral horn of the spinal cord where you have actually the soma of the motor neurons that are affected in this disease. So with very few preclinical data, we filed for orphan drug designation status in April 2021, and we got the orphan drug designation status by the EMA, FDA, and Swissmedic. All data that I will show you were done in the lab of Smita Saxena and conducted by the PhD student Federica Pilotto. So I will walk you through this big slide. It's the rotor-rod experiments. We have done quite some motor performance in vivo models of ALS. One was the C9orf72 model, which is one of the most frequent genetic and mutation variations of the amyotrophic lateral sclerosis disease. You can see on the graphic, you have like on the y-axis, you have the latency to fall on the rotor rod, and on the x-axis, you have the days in postnatal days. So we did a first test with a power number of 6 to 7 mice per group, where you have a treatment of every other day by 15 milligram per kilograms for 10 days. At the early stage of the disease, so between postnatal day 110 and postnatal day 121, and then to an advanced stage between postnatal day 160 and postnatal day 170. We could see that talineuren shows an effect on the motor performance, but as soon as the treatment is stopped, the motor performance goes down again. So we decided to make a continuous treatment and could show that we could, by continuous treatment, starting from postnatal day 110, show the continuous improvement and maintenance of the motor performance, almost as similar as the wild type groups, comparator groups. So we have started with IV, and we followed up with oral. And this study is still ongoing because it's a survival study. The animals get the product in the water, in their drinking water, and we can see similar rescue of the product, basically of the motor performance by talineuren. This is the gray curve on the oral graphic. Basically, you can see like it can reach almost the wild type controlled groups compared to the C9 model untreated. We have then started also to test in another animal model, which is the SOD1 model, which is the other genetic mutation that is regularly occurring in ALS. By IV injection, we could see a similar behavior. We have first tested what happens if we treat for a short period of time, stop the treatment, and we can really see also in that model, we need a continuous treatment in order to preserve motor performance. Now, I will show you histopathological data. I had to restrain a bit of the C9orf72 studies at P170. And then one is a very common protein aggregate, is poly-GA aggregate that can be found in the motor neuron, and talineuren significantly reduced the big poly-GA aggregates in the soma. And then at the cellular level, we could observe a reduction of BIP factor, which is an AR stress marker, as well as the unfolded protein response marker, the ATF4 marker. We could see that the treatment of talineuren basically strongly reduces all these stress factors at the cellular level as well. We did some analysis of the muscle fiber integrity in the C9 model, also at postnatal day 170. And on your left, you can see a H&A staining, and you have like the C9 model on the lower row, where you can clearly see a big difference that the C9 has this typical wandering of the nuclei of the muscle fiber, basically, which could not be observed in the treatment, of C9 with talineuren, compared to the wild type, where none of these events could be found. This is a typical sign of muscle fiber cell death, basically. And another thing that you can observe is like the shrinking. So here you have a perfect integrate in the perfect tissue, whereas here you see that the muscle fiber starts to shrink and you have these white parts around. And on your right, you can see an NADH staining for mitochondria, where you see type 1 and type 2 muscle fibers that are clearly in the wild type cell line and Talineuren very well preserved. And we could also rescue the preservation of the mitochondrial function, basically, with the treatment of Talineuren compared to the untreated samples of the C9 model. So in the overall, that was a snapshot of all the mechanistics and data we analyzed. And in the overall, you will see some green dots which represent Talineuren. We could actually show that it reduces denervation and muscle fiber degeneration, that it can prevent protein aggregation and thus protein misfolding as well. We have also shown in some other studies that it improves and preserves proper functioning of the calcium signaling as well as the mitochondrial dysfunction. And it has been also shown to actually protect the cells from glutamate toxicity. And we have also some data on the proper functioning of microglia and thus also a reduction of the inflammation. So in that overall, and I think in that model, we could really see a nice potential of what Talineuren can do on the in vivo model and also at the ex vivo. And we have analyzed now data from patients, so cell-derived motor neuron, patient-derived motor neuron, to have a look at this translation at the cellular level. And this will be a very big pivotal study to actually start a clinical trial, which we are planning in the coming weeks, actually. So with this, I would like to go for patient access pathways, where we have made as fast as possible clinical trials in order to gather data in Parkinson's with the NEON trial and the coming LIP study, and in ALS, where the trial is in preparation. We have also estimated and evaluated several access and special status in Switzerland. For example, the accelerated approval based on a phase 1 and really good data, we could apply for accelerated approval for the ALS because it's an orphan drug designation. And for Parkinson's, we could, based on good and excellent data in the NEON trial that we are hoping for, we could apply for temporary approval. In that context, for Parkinson's, we are now also evaluating to go the PRIME pathway for prime medicines by the EMA. So it's Europe after NEON trial finalization, and go to the US through the breakthrough therapy program after the end of the LEAP study for Parkinson's, because the breakthrough therapy pathways requires a bit more data than the PRIME in that context. Moreover, we have had quite some requirements and requests actually from medical doctors to have access to the medical form of Talineuren. And we have, together with a pharmacist and treating medical doctors, we have now made it possible via Formula Magistralis to have an available form as oral form, and the IV form is currently under preparation and will be available upon the finalization of the safety study of the NEON trial. So in conclusion, I think Talineuren is has a big potential. We are advancing fast, especially in the clinic, and we are now having a very nice view of the potential of Talineuren on the mechanistic base from the preclinics. And I think the regulations are very favorable and give some options to evaluate on how to access and make it possible that patients can get this efficacious therapy as fast as possible. So, Talineuren can, in conclusion, can be administered orally or intravenously. It is very well tolerated in clinical and preclinical trials. It is available through clinical trials and Formula Magistralis and has the potential to be therapeutically relevant for several neurological disorders. And I would like to add another conclusion, because I started 5 years ago at InnoMedica, and it's about when Talineuren formulation development really started. So it took us about 5 years to make a full formulation development and preclinical data gathering to reach actually clinical trials. And in this time, we had at some point to make a formulation design freeze to make the full translation to the clinics, right? So it took us about 1.5 years, 2 lockdowns and a few bottlenecks to make a full translation from research scale to a full cGMP production. And with this, I really would like to state that that would not have been possible with the team we have at our nano factory. I think it's this secret of having R&D, manufacturing, and everyone under one roof to make these processes so fast in the end. And I think for that, I would like to thank you, the whole team. Also, of course, the medical team and regulatory team, as well as our administration and finance team, because without them, we wouldn't have the money. So yes, I think it's a whole, but we are unified between 2 places. We are in Marly, in Fribourg, in Switzerland, and we have some offices in Bern, and we commute between both. When I started, we were 10, now we are 53, so we had a very nice growing curve. And with this, I also would like to thank our partners and collaborators, because they did an amazing job in supporting us and contributing actually to make the fastest way possible to the patients, because in the end, it's for them that we work, is to actually make a benefit that they can profit out of, that they have their life, a better quality of life. And I really would like to thank you for your attention today. And now I think it's time for questions. I think you are muted, so we don't hear you, Christoph.
Sorry, it's the most frequent question. Sorry. Yeah, congratulations to this enthusiastic talk, Dr. Peitsch, and especially due to the fact that you are addressing 2 unmet medical needs in Parkinson and amyotrophic lateral sclerosis, very severe diseases. So, are there any questions from Professor Barnholz or some?
Yes, I have a few questions. Okay. First of all, really very nice presentation.
Thank you.
And seems to be a very good job, very methodological. I don't know if you are aware, maybe you are, but many, many years ago in Italy, they have a company and they have a scientist, I think his name was Tetamanti, if I remember well. This was really many, many years ago. And they actually were kind of developed GM1 formulation that were sold in Italy, I remember. Are you aware of it?
Yes, I think you mean Kröner by Fidia probably, but this is like pretty a few years ago. But it was not a pure GM1 ganglioside. I think then there were also clinical trials, and there were quite some publication from the 80s and 90s. But that was a mix. It was a ganglioside mix. It was not a pure GM1 product.
Okay, never mind. He told me also about GM1, but I don't really remember very well. But I have some specific question. You said that you compared the mice cells and the liposomes are superior, right?
Yes.
Okay. So, yes. But you didn't say any details about the liposome. I know from the way they look that they must be about 100 nanometer,
Yes. So you would like the physical-chemical properties and morphology. So our liposome has a dynamic light scattering radius of 55 nanometer, and the membrane to membrane diameter by cryo-electron microscopy, it's 30 nanometer.
Okay.
So, like you have this shell probably because of the GM1 as well, that makes it a bit bigger by dynamic light scattering.
That's expected. Based on the length of the GM1. And GM1 usually it used to be from brains of animals, mainly brains of cows, or where and there is a problem with it. So where do you take your GM1?
Good question. Very good question. So we have indeed, it's true, the beginnings of the GM1 and the ganglioside extracts, which also started in Europe by Kröner by Fidia. It was actually bovine extract.
Fidia, that's a company.
Yeah. And then actually that is why also it stopped. So if you check literature and comments, you have a big black hole basically on GM1 and other ganglioside research because of the BSE, right? And then actually there are like some companies that started to make the extracts from other animals, and our extract comes from a Swiss production from porcine brains. And actually the extract is so purified that India is of the regulations. We check, you have so many steps, and it's so pure that it's almost considered as an animal extract, but it's almost a chemical entity because of the new regulations also. Yeah.
Okay.
There have been also discussions to make it synthetically. I think there are like some patents on this. But the problem it's you lose a lot of material. So we would need to basically make a better synthetic pathway to make this possible. And this is not yet our business case, but this is definitely something to be considered.
Very difficult, especially if you want it to be stereo-specific. And David Shapiro, a long time ago in the Weizmann Institute, he died. He was a terrific chemist of sphingolipid and glycosphingolipid. At the time I worked on this system, I remember he actually made the GM1 synthetic, but it was not, he said it's terrible. The losses are huge, but the main problem, it's not stereo-specific.
That's another issue, and the problem is it's not yet scalable, right? If we talk about GMP production and clinical trials and future market applications, this means actually big numbers in terms of kilograms of GM1 we will need, right? So
okay, but now it is possible. I was involved in a project of developing synthesis of stereo-specific sphingosine, ceramides, glucocerebrosidase, so we can go to also to GM1 if there is a need and there is a market. It's a lot of work to develop it, but it can be done. Okay. Do you have any kind of knowledge about the distribution of the GM1 between the two leaflets of the lipid bilayers?
No, that's a good question. We haven't analyzed this in detail yet.
Okay.
Yeah, that's a good question. This is something we are discussing actually if we need and how to make a proper assay out of this. Actually, we had a meeting like a few weeks ago on that, where we discussed the relevance also,
And you actually have I don't know if 2 or 3 ways of administration, oral, sound very tempting, but have a lot of downside that I'm sure you know. Sub-Q.
No, we do IV. IV,
but you also said another way, or I'm wrong?
No, so basically I compared to Jay Schneider's study that he did everything sub-Q, but because of the
Okay.
I forgot. Yeah, the tremendous side effects of the subcute of GM1. Basically, it's almost a soap, right? It's
Really?
an amphipathic molecule, so it's very painful, and patients had to receive subcute amounts of GM1 every day, twice a day, 100 milligram twice a day, in about 5 milliliters of fluids. So it was not marketable, basically, in that sense. Okay. And the IV, the reason for IV is actually the successes of IV application in China. And we thought, well, we will definitely need to think about other ways and roads of applications. But the IV, we thought for the product and the future of the product is the best way to start, because you can also do nice pharmacokinetics. It's also the thing, we know we have very nice proof of concept that it's working via oral application. We can also find the labeling, so the dye of the label in the target tissues via oral applications in animal models, but we don't find it in the blood. So it's almost impossible to make pharmacokinetics. And this is why we thought, okay, we start with IV, and we will see to probably add oral arm after safety assessment with intravenous.
Okay, the problem with oral, especially in human, not in animals so far, because animals are fully controlled, they are in cages, the same condition, temperature, light, so on, everything the same. But human, unfortunately, they are not. So everyone eats differently, and therefore orally is, I don't know how important is the exact dose and the availability. One thing that you have to do is really availability, but you said you cannot by orally detect it in plasma.
Yes, that's true.
So availability, so you cannot test availability.
No, but we find it, that's the point, but we find it in tissues.
Yeah, but in human you cannot study in
no,
tissue. So, I mean, this will not go through the FDA, definitely.
Okay, so I don't want to be unpolite, but do you think it...
No, be, it's okay.
Sorry, Professor Barnholz, but I don't want to be unpolite, but we are a little bit running out of time. So the next... Thank you very much again, Dr. Peitsch. We can continue the discussion afterwards in the debate lounge.
Sure.
So the next scientist who will refer is Dr. Andrew Cavey, and I see talking about phase 3 study in prostate cancer patients, and so the floor is yours, Dr. Cavey.
That's better without the mute. Thank you very much, and thank you for the kind invitation to join the conference. I've been sort of blown away by all the speakers of the last day or two, so it's a real pleasure to be among you today. I'm Andrew Cavey. I work at Novartis, and I lead the prostate cancer programs at Novartis. And today I wanted to talk to you about a new platform in cancer care called radioligand therapy. And I'll talk about its applications in prostate cancer. Just a disclaimer that this is all publicly available information, and the views are my own, not necessarily those of the company. So I'll go through a few things. I'll just give a highlight of what radioligand therapy is, talk about prostate cancer specifically and why there is still a need for new treatments there. And then I'll talk about PSMA-targeted radioligand therapy, which is the kind that's most advanced in development for prostate cancer. And at the end, I'll touch a bit about where we go next from here. So radioligand therapy, there's several different components. The first is identifying on the surface of cancer cells a target to go after, a receptor. We then have the drug, and the drug is composed of a few components here. A radioactive isotope, which does the killing of the cell. That's linked often in a sort of simple chemical cage here, this linker, and that is attached to a peptide that targets the cancer cell. So it's these four components really that work together in this therapeutic platform. And this is how it works. The idea is that you inject the drug intravenously. The drug then finds the receptors on the cancer cells, and it binds to those receptors. wow, I forgot there was this great animation. Here we go. And once the binding happens, just through simple endocytosis, the whole radioactive isotope and the cage get internalized within the cell. And through simple physical properties, the proximity of the radioactive isotope kills or breaks the DNA inside the tumor cell, causing tumor cell death. So that's how we believe it works. And sort of one of the elegant aspects of radioligand therapy is you can just swap the isotopes. And there are some isotopes which can, if you swap, for example, a radioactive gallium-68, you can use that in imaging, PET imaging. So the idea is you can image the patient with the same drug, but changing the radioactive isotope. You identify where all the target cancer cells are. You replace the isotope with an alpha or a beta emitting isotope, and you can kill the cancer. So that's the conceptual theory about how radioligand therapy works. But let me move a bit more into some concrete applications in prostate cancer. Many of you will know, I think everyone knows about prostate cancer. It is an extremely debilitating disease, very common. And once you reach metastatic castration-resistant prostate cancer, there are many different types of prostate cancer. Once it has spread around the body, which it does for most patients who receive a diagnosis of castration-resistant prostate cancer, CRPC, the survival is very poor. And shortly, I'll describe a phase 3 clinical study that was undertaken in prostate cancer. In that patient population, the median survival with patients with very advanced metastatic castration-resistant prostate cancer, in that population, the median survival was approximately 10 months. So there is clearly a need for new treatments. I mentioned survival. That's not the only outcome that we look at. Quality of life in men who suffer from this advanced illness is poor. And there are not many treatment options, either treatments that are very targeted. Many exist today, but they are limited in different patient groups, or they rely on certain biomarkers that are not widely represented amongst prostate cancer patients. So I'll talk to you specifically now about one type of radioligand therapy that targets PSMA. What is PSMA? Well, PSMA is one of those targets on cancer cells. Over 80% of men with prostate cancer express PSMA. Although there's a lot of interpatient and even intra-patient variability on this, but it is widely expressed. It's also found in other cells in the body, salivary glands it's present in, and the duodenum, amongst others, although at 100 to 1,000-fold lower expression levels versus prostate cancer. Novartis is not alone in advancing PSMA-targeted radioligand therapy. There are a variety of approaches applying the same principle and targeting the same PSMA ligand. But the idea for most of these is similar, where you're looking to deliver a radioactive isotope to a prostate cancer cell. By far the most advanced treatment is a drug called Lutetium Psma-617. So it's a Lutetium-177 radio-labeled isotope. And it applies exactly the mechanism that I described in my first couple of slides. In this case, using the beta emitter lutetium, which we bind to PSMA-617, which targets the PSMA receptor expressed in most advanced prostate cancer cells. The good news is there is advanced phase 3 clinical data. So I'll just talk to you for the next few slides about the VISION study. This was a randomized phase 3 study looking at lutetium PSMA and standard of care versus best standard of care alone in patients with PSMA positive metastatic castration-resistant prostate cancer. Note that it was open label, it wasn't blinded, and that's for the simple reason that with radioligand therapy, it's very difficult to make studies blinded, because you can just buy, on Amazon, a Geiger counter and see whether you've received drug or placebo. So for radioligand therapy, as a principle, we always use open labels. This is a schematic of the design, and essentially the patients had very advanced cancer. They all received PET scanning to make sure that they were expressing the PSMA receptors, and that's what you see on the bottom left here. They were PSMA positive on PET scans with a gallium PSMA agent. They had already received multiple lines of standard of care therapy already. So by the time they entered this study, essentially there was not any new treatments available for them. And the patients were randomized on a 2-to-1 basis, either for receiving the drug treatment Lutetium Psma-617 and standard of care, or just the standard of care alone. And then they were followed up for a period of time. This drug is given every 6 weeks for 6 cycles. We measured 2 alternate primary endpoints. So this means that either endpoint could be positive for the overall study to be positive. One was overall survival, which I think is obvious, and the other one is radiographic progression-free survival. So this is seeing whether patients develop new lesions on scanning at regular intervals. And there was a host of secondary endpoints, which I'll discuss. This slide shows the results from the primary endpoints. The primary endpoints were overall survival on the left and radiographic progression-free survival on the right side of the page. And through these Kaplan-Meier curves, you can see that the Lutetium Psma-617 arm, the purple arm, showed a significant improvement over the control arm. On the left, survival had a median of 15 and change months versus 11 or so months in the control arm. And you can see an even greater and more pronounced impact on the radiographic progression-free survival endpoint on the right of the page. So these were the primary endpoints and what really gave solid phase 3 data behind the efficacy of this medicine. I mentioned there were also some key secondary endpoints measured. You see here ORR, disease control rates, and time to first skeletal events, also which were each positive. The time to first symptomatic skeletal events on the right is an interesting one to look at, because symptomatic skeletal events were defined per protocol in a rather objective way, and they're a pretty close surrogate to quality of life and pain, because they measure things like bone fractures. Here you can see the safety profile of the treatment medicine Lutetium Psma-617. There were more frequent adverse events, and on the right of the page, you can see the treatment emergent adverse events that were grade 3 or above, the most frequent ones at least. And you can see here that in the treatment arm, you did see more, and they were mostly ones related to bone marrow impact, which is probably what you'd have expected given the radioactive nature of the therapy. There are also, on the left side of the page, you can see slightly higher discontinuations in the treatment arm. This is a post hoc analysis showing some quality of life and pain parameters. And again, you see the same trend with favorable profile for the treatment arm versus a standard of care arm, indicating that aside from the survival aspects of it, the patient-reported quality of life was also seen to be higher with this medicine. So in this first randomized phase 3 trial, the drug demonstrated extended survival, radiographic progression-free survival, and a delayed deterioration of quality of life and pain. So where do we go next with this relatively new platform? There is already a drug approved in radioligand therapy for rare neuroendocrine tumors. And this drug here, Lutetium Psma, has just received FDA approval in the United States in the indication I just described above. So where to next? Well, firstly, with this drug, Lutetium Psma-617, we are moving it into earlier lines of therapy with new phase 3 studies. So I talked about the VISION study, which really targets these dark blue bars. We've also started a study called PSMA-4, which moves into an earlier setting of metastatic castration-resistant prostate cancer for patients who have not yet received chemotherapy. And that phase 3 enrollment is ongoing. And we've also started a study called PSMA Addition, a phase 3 study as well, in the metastatic hormone-sensitive MHS-PC prostate cancer population, a large population of people with metastatic prostate cancer. And that phase 3 study will read out in 3 to 4 years' time. We're also evaluating even further studies in earlier lines of therapy. But in general, there's a few different approaches scientifically that we can take, all of which both Novartis and many others out there are exploring. I talked about the use of lutetium as a beta emitter, but there is a strong rationale, certainly preclinically and a bit clinically, to use alpha emitters, in particular actinium, as an alternative isotope. PSMA is widely expressed in prostate cancer, but there are other possible targets that we can go after other than PSMA. As we are doing, but there are still other lines of therapy where we can move into earlier and earlier settings, or adjuvant or neoadjuvant settings. And finally, there are reasons to believe that with various other medicines, either pipeline or existing medicines, combination therapies might work, combining radioligand therapy with other agents, for example, some DNA damage repair inhibitors. So there's my overview of radioligand therapy in prostate cancer, and I'd be happy to touch on anything now or later in the breakout group. Thank you.
So, Dr. Cavey, thank you very much for this really interesting talk. Are there any questions from the audience here, from the speakers already? If not, I would like to ask you, how specific are these radioligands to the prostate cancer tissue? This is often the question, how specific are these ligands?
Well, you saw the safety and tolerability profile. Well, first, let me just say, preclinically, we know that PSMA is expressed outside of prostate cancer, especially in the salivary glands, but also in some other tissues like the duodenum. Although the levels of expression are 100 to 1,000-fold different. In clinical practice, as you probably saw in the VISION study, I showed the more frequent and more severe adverse events. We didn't see any salivary toxicity there. If I had shown the lower grade events, you would have seen quite much more widespread dry mouth, for example. That was very commonly reported, although patients in that study and clinicians reported it as very manageable. Okay. The toxicity, though, does become a bigger question in two settings. One is, as we move, these patients had very advanced cancer. But as we move into earlier lines where patients might live 10, 20, 30 years, the long-term impacts on the bone marrow especially become really important clinical questions that we need to address. Bone marrow and kidney. And secondly, if we change isotope, in particular actinium, which is one that's being explored as an alpha emitter, that can have much more salivary gland toxicity. So that's another thing that would be on our mind.
Since the general management is asking me that we should now switch over to debate lounge or debate room 1, I think we should do this. So giving the chance also to the other people to ask you or the other scientific talks here. So we should switch over to debate lounge 1. It's already mentioned also in the chat. Right to the picture, I think.
Okay.
So see you soon.
Yeah, see you soon.
See you soon.